Thank you so much for the introduction, and thanks for the organizers giving me the opportunity to talk here. I also very much agree with Victor's talk about going back to nature in order to target potentially new drugs in chemotherapy. And I kind of want to use this slide to give a very brief overview on my views of where we potentially can find new drugs and where we can potentially find new leads to target new types of diseases. And as you probably all know, traditional drug approaches really start by screening high-throughput libraries, large libraries for a specific target. Major pharmaceutical industries typically use this approach where 10 to 100,000 to 500,000 compounds of known structures are being screened towards a specific target of interest. This obviously gives a lot of restrictions. These molecules have been made, they have been tested against a variety of different diseases. They're typically, following Lipinski's rules, they're typically heterocyclic aromatic molecules. And so you're really not going to find more different types of chemical space and potentially different types of mechanisms and different targets with these molecules. Repurposing is also becoming more and more popular approach to trying to find new drugs for different diseases, and that's mainly driven by the low cost of discovery because a lot of the human data may already be available for those molecules. Patent mining is also a very popular approach by the pharmaceutical industry, where you look at existing patents of molecules. And again, all of these, in my view, have a limitation where you're really just exploring a very narrow window of chemical space for potentially the target of your interest. So what we're trying to do is go back to nature, and we're trying to look not at peptides, but we're focusing on marine sponge metabolites and trying to look out for inspiration for new types of chemical structures. And if we can find new types of chemical structures, our hope is to find new types of targets and also new types of mechanisms to find new leads for different diseases where we need better or improved cures. One example shown back here is a marine sponge metabolite that is actually clinically used in the UK. And of course, this is a molecule that you probably will not find in the typical high-throughput libraries of pharmaceutical companies. So with that, I want to look at a few natural products. So these are some of the natural products we're trying to target in our laboratory for the total synthesis. But I think more importantly, what we're trying to look at these molecules is, can we first find out what their molecular action is in cell culture, trying to find out what their target is, but then make these structures more drug-like. So in the second program in our lab, what we're trying to do is look for the chemical features that are present in all these natural products and try to generate small libraries of these drug-like or natural product-inspired scaffolds. And one of the requirements for these molecules is that we can make those from one starting material, typically in about 1 to 3 steps. So we should be able to access an SAR of these molecules very rapidly, and all of these are templates that are found in these natural products. So our target is then again not a very specific picked protein that we're trying to inhibit. We're looking at a signaling pathway. And Frank Decker already explained the NF-κB signaling pathway yesterday in a very nice fashion. And we're targeting the same pathway. And the target again of that pathway is really not relevant to us. We're trying to target the signaling pathway and then go back to find out what is the target that modulates that pathway. Our disease of choice is multiple myeloma. I'll tell you a little bit of background about multiple myeloma. It's a bone marrow type cancer. You have a differentiation of beta cells, of B cells in your bone marrow. You form these globular type tumors that also will start degrading your bone or your bone tissue, and so you get this Swiss cheese-like skeleton with tumors growing inside of it. It's a very horrible combination. There's no cure for it. There is a treatment for it, which is by targeting of the proteasome, and the reason that this has become really the only strategy to target this relates to its regulation of NF-κB. So really, in short, we have here multiple myeloma cells. NF-κB is constitutively expressed. It induces cytokines, as we saw yesterday, which activates NF-κB in bone marrow stromal cells, which activates osteoblasts, which activates NF-κB to generate more IL-6. And you get this vicious loop that continuously fights resistance and also continuously induces growth of the multiple myeloma cells. as well as degrading the osteoblast and osteoclast in terms of its bone marrow density. So this is the cycle that we're trying to inhibit in the multiple myeloma cells. And the most important thing I think I want to point out is that 97% of patients are currently becoming resistant to treatment within about 3 years of treatment, after which there's only 1 year left for the patient. So this is clearly an area that needs a lot of attention. So the assay that we've set up is an NF-kappa luciferase assay. We do this in HeLa cells. We try to look for inhibition of NF-kappa B regulation. After we've done that, we then look what is the target of that signaling pathway. Once we have found the target in cell culture, we look for confirmation in vitro in the purified protein assay. We do the chemical optimization and then go back to cell culture as well as animal studies. One of the molecules I want to talk about today is this imidazoline, as shown back here. We prepare these imidazolines through a variety of different methods. We can start with amino acids, make oxazolones, and then we can induce a 1,3-dipole cycloaddition reaction to make these types of imidazolines. We have complete control of reacher selectivity and diastereomer selectivity, as we published in these papers, and I won't have time to go over the details of that. We also have control of enantioselectivity. We can start with aziridines, which we can prepare as single enantiomers, and then do a ring expansion of the aziridines to make these types of imidazolines. So I'm showing this slide to really indicate that we have access to any diastereomer selectivity, regioisomer as well as an enantiomer that we so desire. So in our first assay, what we do, we take the HeLa cells, we activate the HeLa cells with a cytokine such as TNF-alpha. This activates the NF-kappa B signaling pathway. NF-kappa B will move to the nucleus, binds to the DNA, and expresses luciferase. And everyone has seen luciferase probably, which is the same what we see in fireflies. They'll just light up. So if we inhibit this pathway, we can actually see an inhibition in the luminescence in these assays. We found that these imidazolines reduced NF-kappa B mediated luciferase expression, which really indicates that we inhibit NF-kappa B mediated gene expression. And we've done a variety of molecules. I'm just showing you 3 back here where we see inhibition of that signaling pathway. Since we're more interested in gene products and trying to see if this could potentially have drug applications, what we do, we take human blood from patients or from volunteers that we then, again, activate with IL-1 beta, which is a cytokine that activates NF-kappa B mediated gene transcription, and then we either measure TNF-alpha production or IL-6 production, because these are 2 cytokines that are really critical in the pathogenesis of multiple myeloma. And we see very nice dose responses in the presence of this molecule, so we know it translates even in a dirty media like human blood. More specifically, in terms of the pathway, what we see here is a representation of how NF-kappa B, which is made up of these subunits, is present in the cytoplasm of a cell. It's bound to this inhibitory protein, and we have antibodies. There are red fluorescent antibodies that will detect this inhibitory protein called I-kappa B. Once we activate this, and that's shown back here. We see these red fluorescents, which indicates the I-kappa B protein in cells. Once we activate this pathway with a cytokine, you get ubiquitinylation of I-kappa B, which marks it for proteasomal degradation. So the proteasome will come, it will degrade the inhibitory protein, thus liberating NF-kappa B, which then moves into the nucleus and induces gene transcription. So what we see within about 10 minutes after we treat the cells with TNF-alpha, we see complete disappearance of the red fluorescence, which indicates that the proteasome has degraded that red fluorescent I-kappa B protein. The blue here is just a DAPI stain of the nucleus to keep track that we're not killing off the cells. In the presence of the imidazoline, what we see here is a 1 micromolar concentration of the imidazoline. After we treat the cells with TNF-alpha, we completely block the degradation of that inhibitory protein. So in terms of the pathway, we again don't have the target identified, but what we see here is the blockage occurs right here prior to the proteasomal degradation of I-kappa B. So this indicates that this might be a proteasome inhibitor. And so we then moved into the proteasome and tried to read a little bit about what the the significance of the proteasome is. Just to give you a short depiction of the proteasome itself, it's this large barrel-shaped structure. It's about a 2.4 megadalton complex protein, so it's a very large structure. And what it does, it has this 19S cap that will recognize, target, or tag proteins. It will grab onto these proteins, it will pull it down this tube, in which it has 3 catalog sites that will start degrading the protein through proteolysis of the peptide backbone. As I mentioned at the beginning of the talk, bortezomib is a proteasome inhibitor. That's currently the leading drug and really the only treatment for multiple myeloma. Bortezomib will bind in the catalog site, in each of these catalog sites, therefore blocking its proteolytic activity. And if it blocks that proteolytic activity, the cells are no longer able to degrade proteins. As I mentioned before, too, 97% of the patients become resistant, and that is because of mutations that are forming in this catalog site that prevent bortezomib from binding to that catalog site, and therefore it will be no longer effective. And that really indicates the problem. So there's no treatment or no cure for multiple myeloma. And that is because all shown structures back here, these are scaffolds shown back here, is going to be taken over from bortezomib in the market. It's already clinically approved as of about 2 months ago. All these other second-generation molecules all operate through the exact same mechanism of action. I listed here the status of these molecules in terms of the clinic, but really what it comes down to, these are all peptide structures that bind in that catalog site of the proteasome. They have an electrophilic warhead in each of these molecules that bind to that catalog threonine and block proteolytic activity. And that's currently this current state of the art, which also means if you get mutations in that catalog site, there's cross-resistance among all of these molecules. And again, that's why there's no treatment or no cure for multiple myeloma. And once you start getting resistance, all these molecules are cross-resistant, and you really have no longer a follow-up treatment for the patient. So we tested our molecule against the proteasome in a purified proteasome assay, and we saw that we inhibited 2 of the 3 catalog sites. The third one we didn't touch. We then repeated the experiment in multiple myeloma cells, and we got a similar pattern. So we're excited that we found the target of that NF-κB signaling pathway. On the other hand, we're also a little bit disappointing because now you're hitting a target that's already validated in the clinic, and that's really, once resistance occurs, that really was a dead end. But we looked a little bit more in terms of the mechanism of this molecule for proteasome inhibition. And that's when we found out that the kinetics of inhibition occurred through a non-competitive type mechanism, which means if it's non-competitive, what that means is, I have a cartoon for this, is that if you just have a substrate binding to an enzyme, the substrate will bind. In the absence of drug, the substrate will bind and it has a certain action. In the presence of a competitive inhibitor, such as bortezomib, it will compete with binding for the catalog site. And again, if mutations happen in that catalog site, then the inhibitor is no longer effective. In terms of a non-competitive mechanism, we see that the inhibitor binds at some other site to the enzyme, and it's completely independent on whether the substrate actually binds to the enzyme or not. And that's a unique type of mechanism in terms of proteasome regulation, and it's also an allosteric type mechanism. But again, the most important part is that it's completely independent of substrate binding. So if mutations occurred in that catalog site of the proteasome, if we bind somewhere else, we should be able to overcome that type of resistance, and that's exactly what we saw in resistant cell lines. So we have several THP1 cell lines, which is a leukemia cell line with different levels of resistance. So if we treat this cell line with bortezomib, which is the wild type back here, you see it's an 8 nanomolar inhibitor. If we then increase the resistance to that cell line, we drop down to 2 micromolar inhibitor, and we can calculate the resistance factor. If we treat our molecule in this cell line, even though it's still micromolar inhibitor, and I'll come back to that in just a little bit, what we do see is absolutely no effect in terms of the resistance of these cells. So we very easily are able to overcome resistance, and that makes sense based on its mechanism because it's in a completely different site. We followed up with this molecule in a tumor model, and we have here, there's a multiple myeloma tumor, we have here the size of the tumor. Here's the untreated control. The red line is bortezomib, and the blue line is our molecule at a very high dose. Again, the potency is still fairly weak. It's a micromolar inhibitor. We add in 150 milligrams twice a day, which is an enormous amount. But again, we see absolutely no toxicity, no weight loss, no liver toxicity with this molecule. But the nice thing is we do see actually a tumor regression. And when we look at the tumors themselves, we see about 2 micromolars that have accumulated inside of the tumors and tumor tissue at the end of the experiment. But again, the potency was not very high yet, so we did an SAR. We made over 200 of these molecules. We're now down in the 100 nanomolar range of these molecules. Again, I'm going to show you that these molecules still readily overcome resistance compared to bortezomib in the different resistant cell lines. We made a variety of these molecules of nanomolar inhibitors. This one is actually not as potent, but it indicated very good pharmacokinetic properties. It was orally available, so this molecule we can now add orally to the mice, and again, we see complete blockage of tumor growth in these mice compared to the bortezomib control, which always has to be given by IV. So I see that my time is up, which brings me to my summary. What I'm trying to show here is that marine sponge metabolites really can serve as a great precursor, or going back to nature can give you a really good starting point to identify new chemical space, new type of molecules. And I think what's important with that is if we start looking at new types of molecules, we can discover new types of mechanisms. And that's especially important when we start looking at areas such as resistance. So with that, I would like to thank you for your attention. I would like to thank my co-workers who have done this in my lab. This is the current group. Here we have our previous members in a group. We've always funded by the NIH or multiple myeloma organizations. Again, I'd like to thank you for your attention, and if there's time, I'll hope to answer a question or two.
Staying on time?
Thanks.
Wonderful. We have time for questions. Sorry. You're getting behind. Very good.
Great story. Thank you very much. I have a question. You greatly optimized your potency for the follow-up compound, right? That 165 compound. Yet when you go to in vivo studies, you still have to dose it at 100 mg per kg, if I saw it.
Yeah, but that was oral. So
okay.
in the previous study, it was twice a day, and it was by IP.
Okay.
In this study, it was done orally. We have not optimized that dosing yet.
Okay.
That was our first study to just. We have no maximum tolerated dose. It was just an amount we tried to work with.
So solubility and permeability are very low for this compound, or?
So the pharmacokinetics of that molecule is very nice. Solubility is up to about 2 milligrams per mil of that molecule, so it's relatively nice. It doesn't have to be completely soluble even when we go and administer it orally. Right. But this molecule was given as a clear solution. Yeah.
Okay. Thanks.
I have a question about your SAR. You say you would like to get it more potent. So from your SAR so far, do you have any clues as to where you have to go to make it more potent based on the previous kinds of antagonists, perhaps?
Yeah, we're still going blind. That's the problem. We still don't have a crystal structure of this particular molecule. We've made about 200 of them. We're kind of stopping with that right now. We have the potency we want for in vivo models. We want to get a crystal structure or binding site characterized first before just to be more rational at that point. Yeah.
Thanks. Anybody else have any questions?
Thanks for the nice presentation. If you target proteasome, you target in principle a very general mechanism, but you say that compounds show very little toxicity.
Yes.
So how can that be explained?
Yeah, that's a great question. So that's the key question, actually. We don't see abrogation of all pathways. And so we don't see inhibition of or accumulation of P53 and other FOXO, other transcription factors or other molecules that are ubiquitinated. We see a subset of proteins that are ubiquitinated starting to accumulate in cells. There seems to be a very strong selectivity, which has to do, in our understanding right now, with the allosteric modulation of the proteasome. So we seem to get selectivity between what proteins we degrade and what we don't degrade. How that works and which one, we're right now doing a full proteomic analysis of a cell to see which ones we target. But we do know we get very strong selectivity. Somehow we got lucky that we hit NF-kappa B and not some of the other ones. So.
Okay, thanks.
So time for one more. Anybody has one? If not, oh, here we go. Thank you.
So these imidazolines, I know that they are quite, let's say, sensitive compounds in the sense that they can be hydrolyzed relatively easy. So how metabolically stable are these compounds? They
are extremely metabolically stable. So we have an ester group as one of the molecules, which is very sterically hindered, and it does not get hydrolyzed by esterases. We've incubated for 48 hours with mouse blood, which is very high in esterases. The imidazoline, but the C2 position actually is pretty hard to hydrolyze, especially if the molecule is as sterically hindered as ours is. We see completely no hydrolysis when we do metabolic studies in vivo.
Okay,
so
great.
Okay. Okay. Thank you very much for a wonderful talk and.